Welcome student. This is Vikas Sampath Varpe, working as Assistant Professor, Department of Zoology in JMD Arts, BW Commerce and Science College Sinnar. In this video lecture, we are going to start antibody classes and their biological activities. Okay, here mainly the 5 classes are present, and these 5 classes are IgM, IgG, IgA, IgE and IgD is here. Okay, the IgM is a heavy chains normally present in the mu heavy chains, and the molecular weight is 900 kilodaltons, and the total percentage of IgM is present near about the 6%. Okay, the fixed complement is present here, and they play the major function is a primary response against to the pathogens, against to the bacteria, against to the viruses also. Okay, this one is a fixed complement, and mostly the monomers serve as the beta cell receptors. The second antibody is here, anti-G antibody. They are also called as the gamma antibody because of the heavy chains present here, the gamma chains normally. Okay, the molecular weight is 150 kilodaltons, and the total antibody serum near about the 80% is here. The highest amount of serum present in our body near about the 80% is IgG. Okay, this one is also show the fixed complement, and they play the major role in the main blood antibodies. Okay, neutralizing toxic substances and opsonizations. Opsonizations means they are easily identified to the antigens to attach of antibody. They have the special type of receptor present on the surface here. Then the next one is the IgA antibody. This one is also called as the alpha antibody. That alpha antibody because of the heavy chains here, the alpha heavy chains. The molecular weight is 385 kilodaltons, and the total percentage of serum in body is only 30% here. Okay, this is the second largest antibody in the human body. This one is a fixed complement, is totally absent here, and they play the main role in the secreted of mucus, secreted of tears, and the secreted of saliva. This is a more sensitive antibody, sensitive antibodies. Okay, then the fourth one is the IgG. It is also called as the epsilon. That means here the epsilon heavy chains is present, and the molecular weight is 200 kilodaltons. The percentage is very small inside the body, just near about the 0.002% only. The fixed complement here totally absent, and they play the major functions here, antibody for the allergic and anti-parasitic activities. Okay, then the next one and last one is the IgD antibody structures here, and these anti-D antibody structures have the delta antibody called as a delta antibody, and this delta antibody have the heavy chains here, the delta heavy chains. The molecular weight is near about the 180 kilodalton molecular weight, and the total serum present inside the body is just the 1% only. This is a fixed complement, is totally absent here, and the main functions here in a beta cell receptors. Okay, then let's see the one by one. Okay, this one is a IgG antibody. Okay, IgG antibody, and this IgG antibody is the most abundant immunoglobulins. Okay, we know that the antibody is also called as the immunoglobulins. Okay, that's why we are called here the immunoglobulins. This immunoglobulins is total serum is 80% in the human body. This 80% serum is only IgG antibody. Okay, here the 10 milligram present in per unit. Okay, IgG1, IgG2, IgG3 and IgG4, it is a decreasing serum concentration. That means the higher percentage IgG1 is present here, and the lower percentage is IgG4 here. Okay, IgG1, IgG3 and IgG4 are able to cross placenta. The only here the IgG2 does not crosses the placenta here. Then the next functions, the IgG3 most effective complementary activator. Okay, Then IgG1 and Ig3 is a higher affinity for the good opsonization. What is the meaning by the opsonization? Opsonization means the ability to the attract of the antigens. Okay. Normally the antibody is attached to the antigen parts. Okay, and this antigen is attracted to the antibody structures, and the opsonization is a very important concept. They are the specifically attract to the antigens here. Okay, this is the structures of IgG antibody. Okay, this is a Y-shaped structure. We know that this structure is known as the Y-shaped structure. This is a Fab region. Okay, and this region is the lower portion is called as a FC regions. Okay, this one is a constant region, and this upper part is a variable region. Okay, this one is a light chain. This short chain is called as a light chain, and this one is a long chain is called as a heavy chains. And in middle side the hinge region is present, and this hinge region is normally the proline rich regions here. Okay, this is a flexible region. Flexibility. They have the flexibility here. Okay, then the next part is the IgM antibody, and these IgM antibodies are near about the 5 to 10% serum in the human body. Okay, just only 1.5 milligram in per ml. Okay, then IgM also IgD expressed on the beta cells as a BCR. BCRs means the beta cell receptors. Okay, this structure is normally called as the pentameric structures. Why called as a pentameric? Because of the here the 5 IgM structure. This one is the first, this one is the second, this one is the third, okay, fourth one here, and the fifth one here, and they are attached to the J chains, and J chains means the joining chains. The joining chains join to this 5 structure, and that's why this structure is called as the pentameric structures. Okay, the first IgG of primary immune response. Okay, this one is to show the primary immune response. Okay, and this one is a high valence IgG. Okay, 10 theoreticals and 5 empiricals. Okay, 10 theoreticals and 5 empiricals here. This is a more efficient than IgG and complementary activity. This is a whole about the IgM structures. Okay, then the next one is the IgA. This IgA are near about in human body 10 to 15% of serum, and this one is a predominant Ig in the secretions of milk. They are mostly present in the milk. Okay, this one is normally present in the female milks, then the present in the saliva, tears, and mucus also. Okay, near about the 5 to 15 gram IgA is released in the secretions. Okay, at a time secretion they show the 5 to 15 gram of IgA. Okay, the serum is mainly the monomeric. There are also polymers possible, not common through the. Okay, then the secretion as dimers, tetramers, J chain polypeptide, secretory complement, and that's why we are called here the poly IgR here. Okay, this is a structural representations. Okay, this one is the one structures of Y-shape antibody, and this is the second structures of Y-shape antibody. Okay, they are the joining here with the help of a joining chain. In a short form, this region is called as a J chain regions. Okay, these J chain regions also show the flexible regions, and here the secretory components are present. Okay, secretory components are present, and they secrete the continuously J chains here. Okay, this is a basic structures of the IgA antibodies. Then the next one is here the IgA antibody transport across the cell, and this concept is known as the transcytosis. The transcytosis is only possible in IgA. Okay, the transcytosis not only possible, but transcytosis also show in the IgM also, IgG also. Okay, here the formations of secretory IgA, in the regions of submucosa. In the submucosa region, here the plasma cells is present, and this type of plasma cells they secrete the dimeric IgA antibody structures, and the surface of the epithelial cell the receptor is present, and this receptor is known as the poly G receptor. This poly G receptor attract to the dimeric IgA antibody, and they are entering to the epithelial cells. The epithelial cells here the normally vesicles are present, and inside the vesicles these complexes are formed here. The complex is called as the dimeric IgA and poly G receptor complexes. Okay, and these complexes the vesicles out of the lumen structures in human. Okay, and this one is a secretory IgA structures. Okay, and this whole process is known as a transcytosis processes. Then the next part is the IgE parts. This IgE is a very low serum concentration, and this near about 0.3 microgram to per ml. This one is a participate to the immediate hypersensitivations reactions. There are the asthma, anaphylaxis, hives, etc. Okay, and they are bind to the mast cell and blood basophils in Fc epsilon regions. This one is the Fc epsilon regions, and this Fc epsilon region the bind mast cells are here. The another functions here binding the causes of the degranulations, histamines. Okay, histamines. This is a brief introduction of IgE. Next one is antibody IgD. This IgD express the beta cell surface. Okay, and this IgM and IgD express the beta cell surface. These are the two. Okay, IgM is also expressed here, and IgD is also expressed here. We do not know any other biological effector activities. Low serum concentrations is only 30 microgram in per ml. This is the structures of IgD antibody. This one is a Y-shaped structure. Here the light chain is present, and here the heavy chain is present. It is a look like as a just Y-shaped structures here, and the Ig look like as a IgG antibodies. Okay, then this one is our table representations, and in this table representations here the 5 classes is present. Okay, IgG, IgM, IgD, IgA, and IgE. Okay, normally the general structure of IgG is a monomers. Okay, this is a just Y-shaped structure, that's why we call it as a monomeric structure. Okay, the general structure of IgM is a pentameric. The 5 sites are here, 1 2 3 4 5 sites are here. Then the IgG structure is also show the monomeric structure, just a Y-shaped structure. It is difficult to identify IgG and IgD. They are look like as the same. Okay. Then the IgA is a dimers. Okay. This one is the first antibody, second antibody, and they are joined to the J chain, joining chains. Okay. Last one is IgE is also monomeric. Here the 3 monomeric classes. One is IgG, IgD, and IgG here. The location of IgG is free in plasma near about the 80% of circulating antibody. Okay, normally they are present in the blood. Then the IgM is a surface of beta cell free in placenta, and IgD is present in also beta cells. Okay, IgM and IgD present in the beta cells. Then IgA is the monomers found in the plasma, polymers, saliva, tears, milk, and other body secretions. And IgG is secreted by plasma cells, skin, and tissue lining, gastrointestinal and respiratory tracts. The functions of monomers is abundant antibody in primary and secondary response. Okay, cross the placenta and provide the passive immunization to the fetus. The IgM play the major role in an antigen receptor on beta cell membrane. First class of antibody released by the beta cell during the primary response. The IgD show the functions here, cell surface receptor of mature beta cells. Okay, this is the important in beta cell activations. The IgA show the protect the mucous surface, prevent the attachment pathogen to epithelial cells, and the most important functions of IgG here, they found on mast cell basophils. When bound to the antigen, triggers release of histamine from the mast cell or basophil that contribute to the inflammations of some allergic responses. This all are the functions of these 5 classes. Okay, thank you so much.